Okay, so thanks to Rich for the overview of mucolipidosis 2 and 3 and 2 and for the update on how ML3 alpha, beta, and gamma are related to each other. So I'm going to talk to you a little bit about mucolipidosis, just in terms of the pathophysiology, just very briefly. So it's caused by deficiency of an enzyme that's gluconic-1 phosphotransferase, which we can abbreviate PTAs. And Rich went over this as well. It has alpha and beta subunits that are encoded by GNPTAB. This is not a lysosomal enzyme, right? So you can't just replace it with enzyme replacement therapy. It lives in the Golgi apparatus and is essentially responsible for mannose-6 phosphorylation of your lysosomal enzymes. And we talked about how there's a severe or I-cell or ML2 form and attenuated forms, ML3 alpha beta, and then ML3 gamma, which is encoded by a different gene, GNPTG. The S1, S3 form of the PTAs is a truncated form, so it does not occur in nature, but it has elements of the native gluconic-1 phosphotransferase. It's smaller, it fits more readily and easily into the AAV plasmid, and is more efficient at phosphorylating lysosomal proteins. It also doesn't rely on the gamma subunit to phosphorylate lysosomal proteins. So this was put into AAV. This was done by M6P Therapeutics under a CAG or a chicken beta actin promoter. And these were encapsulated into AAV9. These vectors were administered into an ML2 mouse model that we had with the Kornfeld lab. This is a knockout for GNPTAB. And we administered this gene therapy intravenously at 2 × 10^11 into the tail vein at 4 weeks of age in the mice. So in ML2 disease, you see very high levels of circulating lysosomal enzymes, but these lysosomal enzymes lack mannose-6 phosphate. These graphs show an assay using the mannose-6 phosphate receptor to bind the enzymes and then see, how much essentially see how much mannose-6 phosphorylated enzyme is circulating in the serum. So on the, which one is the pointer? But on the left, you see the beta hexosaminidase, B is alpha manosidase, C is beta manosidase. Then the black dots and line at the bottom is pre-treatment serum. And then in the ML2 mice, post-treatment at 2, 6, 10, and 14 weeks with elevated levels of mannose-6 phosphorylated enzyme compared to the ML2 mice. In D is this same data graphed slightly differently. So on the x-axis, you see beta hexosaminidase, alpha manosidase, beta manosidase. The wild type mice are depicted in gray. It's maybe a little hard to see, but there's very low levels of enzyme in the wild type mice that are circulating in the blood versus what you see again in the ML2 mice. The ML2 mice have elevated levels of lysosomal enzymes, but they lack mannose-6 phosphate. So the pre-treatment serum, you can see the bars, for the black bars, are very low at basically the 0 line. And then all of the post-treatment serum levels for the ML2 mice that were treated with the gene therapy, they retain having very elevated lysosomal enzymes in serum, but those lysosomal enzymes have mannose-6 phosphate on them now. Okay. This is a depiction of the expression of S1, S3 in tissues in the treated mice. On the left are bar graphs, 14 weeks post-dose of the mRNA levels of S1, S3 in liver, heart, brain, pancreas, and salivary gland. The y-axes vary according to tissue. So you have to look at those as you can see. There's quite a lot in heart. There's a good amount in liver. Pancreas and salivary gland had some. Brain levels were basically the same as carrier as untreated mice. So we saw, even with AAV9 administered intravenously, we saw very low expression in the brain of these mice. On the right is in situ hybridization using the RNA, basically RNA fish-ish for RNA. So you can see in liver and the brown adipose tissue near the salivary gland at 50 weeks, you can still see good expression of S1, S3 in the treated mice. This is histology. This was done by Peter Vogel at St. Jude, as was the ISH that I showed of the submandibular salivary gland, parotid salivary gland, and pancreas. These are tissues that show a lot of vacuolization and storage in the mouse model of ML2. So the left column is the normal wild type mouse. In the middle column, the knockout untreated mouse, and then on the right is knockout treated mouse. And you can see in the untreated knockout mouse, you can see the vacuolization. And in the treated mice, we saw potentially slightly less vacuolization in the pancreas of those mice. Behavior and visual acuity, we did see some behavioral deficits in the ML2 mouse. We studied them at 5 months and at 9 months. We saw some mild improvement in behavior. Notably, they had a slightly improved time on the inverted screen and the accelerated rod, and slightly improved visual acuity. The mouse model, interestingly, has quite a lot of retinopathy and vision loss, which is not really part of the human phenotype. But we do see that in the mice, and we saw some improvement at 5 months with gene therapy. We measured length, weight, and we measured bone mineral density and content with DEXA in the mice. We did not see substantial differences in the bone mineral density and content. Even between the wild type and the ML2 mice, it was really not very pronounced. However, we did not see any differences with the treatment compared to the untreated ML2 mice in terms of weight, length, bone mineral density, or content. We did see a statistically significant increase in survival of approximately 12 weeks. So the red line are the untreated ML2 mice. The green line is the treated ML2 mice. And then the purple and blue lines are the heterozygous and wild type mice. So after this experiment, we concluded that we showed long-term expression. We restored mannose-6-phosphate content of serum lysosomal enzymes, partially improved pathology of the condition. We hypothesized that the incomplete correction of the phenotype might be due to later initiation of gene therapy, as well as that we could potentially improve expression in the brain if we tried intracerebroventricular administration. So we next started a pilot to study intracerebroventricular injection in the neonatal mice. We first administered 4.5 times e to the 9th vector genomes of the AAV9 with S1, S3 into the mice, and it definitely improved the expression of S1, S3. So I've reproduced that same graph you saw before on the bottom left from the intravenous injection of the gene therapy at 4 weeks of age. If you focus on the brain and look at the y-axis, you can see basically no expression in the brain. On the right is the graph of 2 mice treated with S1, S3 intracerebroventricularly at birth with up to 0.5 to 1 S1, S3 copies per cell in the brain after that. We then looked at neonatal treatment with the intravenous AAV9 S1, S3. The mice did not tolerate a dose of 1 times e to the 11th, so we decreased to a 10-fold lower dose, and they did tolerate that. We then wondered, because we had to go down in the intravenous dose, if it would be better to give the intravenous dose at 4 weeks while giving that ICV dose at birth. And the reason we wanted to give that ICV dose at birth was because at least in mouse models, behavior really only improves dramatically when you're able to administer treatment at birth across lysosomal diseases. So we did a study to determine whether mice treated with ICV, S1, S3, AAV9, S1, S3 at birth would show an anti-capsid antibodies against the AAV9 that might preclude dosing intravenously. We know that neonatal mice tend to be tolerant to things that are administered in that period, but there really haven't been a lot of studies looking at whether those mice can be subsequently dosed after ICV or other kind of neonatal exposure to virus. So here in this graph, we brought the anti-AAV9 capsid antibody titer assay into our lab, and we studied mice that were treated with vehicle, mice that were treated at birth. At FB means from birth, but it's really at birth with the AAV9 S1, S3, and then mice that were treated with that plus intravenous AAV9 S1, S3 at 4 weeks. So the mice that were treated intravenously, you can see at 8 weeks, the red triangles, they do have titers, but the mice that were treated with ICV gene therapy at birth, at 4 weeks when we would be dosing them intravenously, had no titers, and that was maintained at 8 and 16 weeks, such that we don't think that ICV treatment of that AAV9 at birth results in a titer. So because of that, we subsequently studied whether early ICV plus delayed IV or early ICV plus early IV, which would be better in restoring the serum lysosomal enzymes, which would be the better therapy to take forward. So this is the same assay that I showed before, looking at the mannose-6-phosphate receptor binding of mannose-6 phosphorylated enzymes using mannose-6-phosphate receptor with alpha mannosidase, beta hexosaminidase, and beta mannosidase. The blue circles and squares, and I think those are triangles, I can barely see them, represent mice that are treated ICV plus IV at birth, whereas the inverted red triangle, orangish red triangles are ICV treatment at birth plus IV at 4 weeks. So it looked like we did not see restoration of mannose-6-phosphate on lysosomal enzymes when we treated IV plus ICV at birth, and so that delayed IV therapy, because we were able probably to give that 10-fold higher dose, was more effective. So at this stage, we have a cohort of mice that are being treated with ICV at birth plus IV at 4 weeks of age. This is an ongoing experiment. We have enrolled 16 wild-type mice, 14 ML2 mice, and 10 ML2 mice that have been treated in this manner. The mice are presently aged 6 to 53 weeks, so we are in the process of studying them with behavior assays, bone mineral density and content, and survival. We've also, if it's of interest, we've crossed this ML2 mice onto a LAMP1 triple flag line to enable isolation, immunoprecipitation of lysosomes. The goal is in that mouse line that we can evaluate the content of lysosomes in treated and untreated mice to see if we're able to restore the lysosomal enzymes content with therapy. So we're currently collecting wild type and ML2 mice for proteomics and lysosomal enzyme assays to study lysosomal content in different tissues, and then we'll be piloting that as well. And on the right here, this is just a Western blot showing, this is from the Sardiello lab, showing the purification of lysosomes using this LAMP1 flag. Tag is pretty good in most tissues, particularly in the brain and in the kidney. And I think that's my last slide. I'll acknowledge the many people, our collaborators at M6P Therapeutics, Stuart Kornfeld, and research support from the National Institutes of Health, National MPS Society, Yash Gandhi, and I already acknowledged M6P. Thank you.
Patty, thanks for sharing that really exciting work. Let me ask a very naive question. When you said the neonatal mice wouldn't tolerate the IV injection, was that a question just because they're so tiny of the volume you could inject, or was it to do with toxic effects of the vector, or what exactly did you see?
Yeah, they all died.
Okay, well, that's pretty clear.
But it was not it's not a volume issue.
So do you have any idea what the mechanism is?
I do not.
Okay, good. Okay. Do we have further questions from the floor or from our online audience? Yeah, please, Jack.
I guess I'm no scientist, so I don't understand everything, but is this then going to be more like for like like newborn babies? Like if this does work, would this work for children that were older? And would it work for like ML3, or is it just more, or is it more toward the more severe form of the disease with ML2?
Yeah. So, similarly to the wild type GNPTAB, this S1S3 phosphotransferase is simply a replacement therapy for GNPTAB. So it, in theory, should do work as well as the wild type, but for ML2 and ML3 alpha beta. The one thing about S1S3 is that it will phosphorylate lysosomal enzymes in the absence of gamma. So our plan is to test in gamma cells whether that S1S3 will also correct the defect in gamma. But we don't have any results or anything. It's a hypothetical at this point.
But I guess the other point worth making is these are knockout mice. So these are mice that have a very severe phenotype, and which is an appropriate model. But of course, when you talk about translating that to humans, there are a number of challenges, but the same principle applies, which we've been talking about, which Dr. Desnik emphasized, and I think we all know, and that is the earlier we make the diagnosis and institute therapy, the more likely we'll get a better outcome, because no therapy at the moment replaces neurons that have been lost.
Yeah.
And that's the challenge of neurological involvement, because although we've known for 50 years there are stem cells in the brain, there aren't enough to renew neurons that have been lost. And that's why this is the most recalcitrant aspect of treatment.
Yeah. I mean, in terms of the other mutations that might that might result in some protein being produced, right? And so the question is, because the alpha beta subunits dimerize, whether there would be any kind of heterodimer formation or something that might make that that therapy less effective. And it is something we plan to test, probably in human cells would be the best place to test that. So that's one of what we proposed in our next funding cycle. But Stuart's answer to that has always been, well, the heterozygous parents also have one mutant and one wild type, and it seems to function.
Can I just can I reinforce a couple things that Patty just talked about? And one, the fact that the S1 S3 doesn't oh, thanks for the video, doesn't require the gamma. I think that's a huge advantage with this one, because I think initially, thoughts about gene therapy as this approach, the question was, well, would you have to overexpress GNPTAB using a gene therapy, but also have an overexpression of G, of the gamma subunit, considering how this enzyme comes together in a complex. So that, plus the smaller size and easier packaging into vectors, I think are two really critical advantages of that S1, S3 construct. And then, not to say the third, the most important probably being that it's just better at phosphorylating all types of lysosomal hydrolases due to removing those sequences. So those were, I think, worthwhile points to consider.
Thank you.
Thank you, Rich. Any other questions or comments at this point? Okay. Well, again, thank you for sharing that beautiful work and for joining us in person, Patty. So I think at this point, I'm going to invite Jackie back up to the podium.
Okay, so I thought this was really interesting. So thanks, Carolyn, and to everyone that, put these questions together, because I think they really ask some great questions. What the patients deal with and what we as caregivers have to deal with. Obviously, it did not surprise me that I can never say it. musculoskeletal disorders would be the leading issue. I know for sure in my daughter they have been. So it looks like almost half basically have said this. Neurological decline, definitely. We see that especially with our older children as they kind of approach their 30s, especially we see behavioral issues. I think, Susan, we've seen that with the boys specifically, like more so. We're not sure why that is, but you can see a child or an adult go from being really sweet and whatever, and then they change completely, and those are all due to the neurological decline. The organ dysfunction, that's obviously a huge problem. It's what we lose a lot of our kids to. So, but again, when you're living with it, it's, the bone, the joints, and then the neurological decline. So these are the most common symptoms in ML2 and 3 alpha beta. And then I think there was another here that was, did we have oh, hold on, I guess I could hit this. Yeah, there we go. So the burden, yeah. Oh, did I? Oh, we pressed it at the same time. So the desire for improvement, we've got, again, neurological decline being the biggest on this. I think this is probably coming from, a few of us that have the older children. And then we have for the musculoskeletal disorders, that would be second. And then access to care, health system improvements. Somebody touched on that a little bit before. We're lucky. I live in the St. Louis area. I have Washington University. So, and they've been fantastic, but not everybody has an institution right in their backyard like that. And I think it's been really hard for many, many patients and their caregivers to find adequate care. And also, a lot of them go undiagnosed for way too long too. So that's a big one. And then, of course, genetic and enzyme-based therapies, that's on there too. And we are beginning to see something happen in that world now, as Patty just spoke about and what I spoke about earlier. Okay, burden of disease. Burden on patients, the physical limitations is probably the biggest issue. I can totally understand that. My daughter's, and again, these conditions vary so much. We have ML3 patients walking, living a fairly, normal, well, not normal, it's not normal, but they're able to go on to college, university. In my daughter's case, she is very restricted. She's in a wheelchair full time. She had learning disabilities. So all of that has been a big burden on her. I'm trying to read it. I'm terrible. My eyesight. Emotional impact. Yeah. So again, they are impacted emotionally by this. For my daughter specifically, I think the thing that I saw is when her friends at school all began to, kind of catch up with her, go further than her. A lot of them now are married and have children, and she sees that. And there is definitely a sense of grief, I think, for her and for us too. Going to the burden on the caregiver, the emotional impact, it is really hard. Like you're trying to work, you're trying to live, and you're constantly, I think depending on where you are, the help that you can get, for carers and people to come in, a lot of the time, you don't get the help that I think you need. And I wanted to mention, I forgot to say this in my speech, one of the things that I was told when Anna was first diagnosed, I met Denise Crompton. Some of you may know her. And the thing that she told me was the one piece of advice I can give you is that you take care of yourself. Because if you don't take care of yourself, you cannot take care of your daughter. And again, I think that leans in with the emotional impact. You have to take care of yourself as a carer. You can't just let it all come on you. If you do, you're just going to fall apart. Physical burden is also hard. I think it depends on the significance of the disease and where it's at. For Anna, yeah, I'm having to carry a 100-pound young lady around and, bathe her, dress her, that type of thing. Some of the kids are able to do their own thing. ML2, the more severe spectrum, it's 100% all the time. I mean, you 24/7. And then burden on siblings. I have a son. He's 34 now. He was 4 when Anna was born. And I did see a toll on him emotionally. He would get angry sometimes because we couldn't give him the time that we had given him previously. And a lot of it was doctor's appointments and then Anna wasn't too bad. She was able to live a fairly normal childhood until she was about 12 when things really declined. But it had an emotional impact on him. So I think that survey is completely correct. And yeah, it says it on there, reduced parent time. That's definitely something that unfortunately the siblings will see however hard you try. And you can't, I've talked to my son since then and he understands, he understood, but it was hard. Okay, and the barriers, we've talked on this too, lack of medical expertise and access to specialists. Again, I think it depends on where you live. If you're lucky that you live in a wonderful place that has like the universities like WashU, we've had people we've had a couple of doctors that really haven't known what on earth it is that she has, but the majority of them do. And that's really where she was diagnosed, although she was technically misdiagnosed at the beginning. The lack of treatment options, that's also a big plus. And I think that also falls into like medical insurance too, because they're constantly butting heads with insurance here in this country that refuse to cover something because they think you don't need it. And even though the doctor says you do. And then the financial barriers. For us, it was harder when Anna was a child because when they're 18 here, they're recognized as an adult and they can qualify for Medicaid and some of these other things. But it was tough. Like if we needed a wheelchair and the insurance company were kind of fighting us on it. And we ended up having to pay out of pocket for quite a few things too. I think that's it. That's it.
Jackie, thank you. I'm going to ask you if you wouldn't mind to stay here to offer some expert commentary.
Okay.
Because my final task of the
Okay.
meeting is just to talk about potential outcome measures. And I think the message I was hearing is that neurological function is a big part of the burden of disease here. And it seems that mobility is a big part of that.
Right.
I think the mobility really reflects a combination of neurological dysfunction as well as musculoskeletal, particularly joint dysfunction in this disease. And so when we think about potential outcome measures, we come back again to something like the 6-minute walk test, which is well standardized. It's recognized by regulators and would be a reasonable measure, I think, of people's function. Mm-hmm. Would you agree with that?
Would. They did do that with Anna, back when she was able to walk. And, that was one of the measures that they did early on. So I think that's important.
There are other measures too, because of course the hand involvement is very prominent. Yes. ML2 and 3. There are a variety of fine motor scales that could be used to assess that. Right. In terms of joint involvement, there can be some relatively simple things, just measuring range of movement, mm-hmm. which has been used in some studies.
Yeah, Anna was, and that's how we first found out that Anna had mucolipidosis, because she was doing ballet, and this is as far as it would go. Yeah, that was it. And her fingers were curled. And that was what when we went to Washington University, the diagnostic doctor that we saw immediately, he thought it was MPS. Yeah. But when we got the blood work done and everything, we found out that it was ML. But that's definitely something.
Fair enough. And again, perhaps for some of the younger children less affected, there are some other tools that can be used, such as gross motor function scales, which have been used, other more general scales, such as the WeFims, which can be used as a more general outcome, which is well known for functional impairment. I think the other issue that comes up is that you've raised, and we've heard with other disorders, are the cognitive function, which of course we can measure with age-appropriate instruments. The other challenge, I think, is the behavioral change, which you highlighted, which we see in so many of these disorders. There are behavioral rating scales for those, although part of the challenge is people who have severe behavioral problems are not likely to be very interested in participating in these sort of scales.
No, sure, right, Susan.
But certainly something we could talk about. And of course, where you have organomegaly,
Yeah.
that's something we can fairly easily quantitate. And as we've mentioned before, in these disorders, you can measure oligosaccharides in the urine and similar,
Right.
which give you another outcome measure. So I think we have a number of tools there. Any other aspects you think we haven't covered that would be worth thinking about in terms of outcome measures that would be meaningful from your point of view?
I think you've covered most of them. I mean, again, it's so different in each child, which
Yes.
makes it really hard to determine, you know, the outcome. Like some gone to previous conferences, and some mucolipidosis that are now adults almost seem relatively like I would have a hard time telling that they had the disease, and then others right away I can tell. You know, the
Yes.
facial coarseness
Right.
is more prominent in some. I mean, I think that's one way of definitely telling if it's, you know, whether it be MPS or ML, you can really see.
Exactly. And I think that this is something that Dr. Desnick highlighted earlier, and there's now software utilizing artificial intelligence so you can characterize what some people now call the characteristic facies of these disorders, and can actually get some quantitation there. So that would be a nice non-invasive tool, which could be useful diagnostically as well as in terms of follow-up.
Yeah, at least to let you, like, point you in the right direction that this is probably a lysosomal storage disease.
Yes.
So, and then I think that would be, like you say, very, it's not invasive at all.
Right.
And I know, like, my Anna and I know other ML kids, they don't want to get stuck.
No.
They don't want to have anything that's going to be uncomfortable. That would be a great way to do it.
Yeah.
Yeah.
Great. Well, other suggestions from our in-person audience or online audience about outcome measures? Again, this will be further refined, and I hope we'll have participation from our real experts when we put the paper together. As I say, you know, the experts are the people who live with this disease 24 hours a day, 365 days a year. So we really appreciate your input and insights, because textbooks are synthetic based on secondhand experience. So that real world experience is terrifically important. So Jackie, thank you for your participation.
Thank you all.
Very much appreciated. So